Hi, everybody. My name is Julie Gonzalez-Morales, and today I'll be talking over my project named Digital Twins of Cells Using Computational Machine Learning, and I am working underneath Dr. David Long. Next slide, please. So just some overview is that this research, the overall goal of this research is that it will lead the way into studying healthy attached endothelial cells and diseased endothelial cells and how they interact and communicate with one another and other cells in the body. So specifically, we want to look more closely into the microvascular system and its role in cancer and wound healing. And so just overall, the goal is to eventually inform the study of healthy and diseased endothelial cells. And so just some key terms to help you follow along in my presentation are adherent endothelial cells, which are cells that grow while attached to the culture vessel that mimic the way that they are in the body. And then machine learning. It's a type of artificial intelligence that allows software to become more accurate at predicting outcomes the more it's trained. And so just deciphering, I'm going to be talking about two machine learnings in my presentation. One will be referred to as digital twin machine learning, and the other will be automatic segmentation machine learning. Next slide, please. And so here we can see how we have a multicellular image with the membrane stained in red, the nucleus stained in blue, and focal adhesion stained in green. And so from this image, we can see that we have quite a bit of information given to us in terms of the subcellular features. Next slide, please. And so now when we take away the nucleus stain or the blue, we have even less information about the cell. Next slide, please. And so when we do it again, we can only see the stained membrane of the cells in red. So that's even less information than we had in the previous two images. Next slide. So our ultimate goal is to be able to take the membrane as our only input and be able to predict a 3D digital twin with all of its subcellular features. So the process currently of staining each structure is time-consuming and costly. So eliminating steps would save time and resources. Developing a digital twin would allow for both the morphology and signaling dynamics of a cell to be simultaneously studied, which can't be done with our current methods. So our question is, can we make a 3D prediction from only having the membrane as an input? So eventually, we want to be able to look at signaling pathways in conjunction with morphology changes through this digital twin. But first, the cell morphology has to be predicted with accuracy. And so we want to do this by only using the membrane as an input. So the first step to produce this 3D digital twin is through segmenting the monolayer images first with accuracy. Next slide. So now here's a representative image with the membrane stained. It's turned into single cell images, and then these single cell images that have been segmented, that have the membrane segmented, that resulted from the automated machine learning are then fed into this trained machine learning that will predict a digital twin. And so then the morphology of that cell will be predicted with only the input of a membrane. And so the reason why this multi-cell image is being turned into single cell images is due to its quicker processing time and the fact that better results were yielded when we did it this way. Next slide, please. A key step in this process is the automatic segmentation. And so just diving deeper into this, a separate machine learning from the digital twin is trained to automatically segment these raw images that the machine learning has not seen before. And so the raw endothelial monolayer that is segmented automatically is then split into individual cells, and then those are used to train the digital twin machine learning. Next slide. So for example, here we have an image of a endothelial monolayer, and we have two manual segmentations done by two different people. So here, there's quite a bit of disagreement there. And so by automating this process, it reduces that manual error or manual subjectivity. So for the same input, you would receive the same automatic segmentation each time, no matter who ran the endothelial monolayer first. Next slide. So we are still in the phase of training this machine learning digital twin, which has kind of been the bottleneck of this project. And so it was unanticipated, the heavy focus that was switched from producing a digital twin to getting this automatic segmentation to work. So right now we're at a point where we have to be able to automatically segment each cell from this multi-cell image instead of doing it manually for the reasons I previously explained. And so the reason for this is so there is no disagreement or gray area introduced between what is part of the cell membrane and what isn't. And so being able to do this portion automatically would yield the same result each time when using the machine learning to automate this cell segmentation. Next slide. And so the way that we're automating this process, like I mentioned before, is by machine learning. And so we are collaborating with Dr. Jianqiu Chen and Justin Sonic from the Leibniz Institute of Analytical Sciences in order to do this automatic segmentation. And so in order to do this, we need many images to train the machine learning. And so that is what we're currently working on. Next slide, please. And so here we just have some preliminary results of the automatic segmentation from the machine learning. And so as seen above, you can see there are discrepancies between the manual segmentation on the right and the automatic segmentation in the middle. And so theoretically, the more trained the automated segmentation machine learning is, the better the segmentation should be. Next slide, please. And so just taking a step back, we essentially want to be able to automatically segment the membrane of the cells and place each segmented cell into single cell images, feed those into the machine learning that has already been trained to produce a digital twin, and then obtain that prediction for each cell. And so once that prediction has been completed, we want to reassemble the image we started with, but this time it would have all the predicted structures such as the nucleus, the F actin, and the focal adhesions. So it would have all the structures and then the membrane that we input in the beginning. Next slide. So just some limitations that we noticed while going through this project are, for one, the focus switched to the segmentation. And like I mentioned before, it was due to that manual segmentation subjectivity, where if two people segmented, the same raw image, they would get different manual segmentations. And so another thing that we ran into was determining the threshold for the machine learning. So when is it considered enough training for the machine learning to produce an accurate segmentation? And so just some future research, we are looking into identifying and extending these methods to new structures in the cell, as well as being able to extend these methods to any adherent cell, not just endothelial cells. And so once the digital twin has been developed, we want to eventually study live cell signaling dynamics and morphology. And so just overall, the importance of this digital twin would be to acquire detailed morphology without having to physically label all of these structures, saving time and resources. And so the more these machine learnings are trained, the better these segmentations and predictions should be. Next slide. Here are my references. Next slide. And I just want to say thank you to the McNair Scholars Program and the KMB program for supporting and funding this project, as well as Dr. David Long and Rex Haffenstein for their guidance and mentorship throughout this project, as well as giving me the opportunity to research in the lab. Next slide. That concludes my presentation. I am open to questions. This is something that we currently have not looked into, but because the machine learning algorithms are trained based off of set of raw images, potentially if you were to train the machine learnings with that in the beginning, then there is a potential chance that you could predict the subcellular features of prokaryotes.